So we finished up with our cell biology. Do you have any doubts with cell biology chapter?
No.
All clear?
Yeah.
Yeah. Okay. So let's start with molecular biology. Can you hear me?
Yeah.
Can you be a bit louder?
Hello?
Yeah. Okay. So we're going to do with molecular biology. Okay. Could you tell me up till where have you finished with molecular biology in your school?
Well, we're done DNA and RNA replication.
Hmm. Okay.
Yeah.
All that, right? Okay. So we're going to start that. So molecular biology. Let's start the chapter. Just a moment. Okay. So molecular biology, when we're talking about molecular biology, here we are dealing with the molecules which are present inside the cell. Sana, am I audible?
Yeah.
Yeah, okay. Molecules which are present inside the cell. You know, every cell, we have millions and millions of cells. So every cell will have their own metabolism. Every cell will have their own biomolecules installed. Okay, so in this chapter we are going to basically study about those. So now when we are studying about biomolecules, okay, first of all, when we are doing microbiology, we need to study something about the biomolecules which are present in our body. Okay, and the most important biomolecules, let us see here. Biomolecules, these are molecules which are present inside the cell. They are being produced inside the cell. Okay, so we need to study when we study the biomolecule, some of the important biomolecules that we are going to study is carbohydrates. We're going to study lipids, and we are going to study proteins. These are the three important biomolecules that we need to know, and they find, they are, they play a very important role in the growth of the body. Okay, in the development of the body, in carrying out an efficient defense system. That means overall growth and development of the body. So we're going to see what are they. Okay, now when we are talking about any biomolecules or any of these, the only thing that comes to our mind is these all are what? They all are carbon compounds. Okay, they all are carbon compounds. That means compounds which are made up of carbon. Carbohydrate contains carbon, lipids contain carbon, and even proteins contain carbon. So carbon is an integral part of every biomolecule that we are going to study. Okay, so we are now going to study on how, and but one important thing that you need to know is that these carbons which are present in carbohydrates, carbons in lipids, carbons in proteins, they are differentially arranged. They're not arranged in one similar manner, they are differentially arranged. Okay, so what I'm going to do, Sana, is that we are going to study about each of them. Okay, now I'm going to go into the detail of carbohydrates, polysaccharides, everything, lipids, proteins. When I do proteins, we are going to also study about a very important concept that is we call as Ornithine cycle. Okay, we are going to study about Ornithine cycle. All right, then we are going to study about water, the important component of life. After that, I'm going to go into the metabolism. Okay, I'm going to teach you in a very sequential manner. Okay, metabolism, we're going to see on metabolism. All right, different types of metabolism we're going to see on that. Finally, we are going to check out on the enzymes. Okay, we are going to check out on the enzymes, and then we are going to also, after enzymes, we are going to see on the nucleic acid, nucleic acids. Nucleic acids, we are going to see DNA, and we are going to see RNA. Is okay? All right. Is this clear? Yes.
Yeah.
Yes. Any doubt?
No.
Yes. And then, of course, when we study the nucleic acids itself, we are going to study about the transcription, translation, and all other different aspects which are involved in the in especially in molecular biology. Have you done transcription, Sana?
Yeah.
Yes. So we're going to go into transcription. We're going to go into cell respiration, photosynthesis, everything. But first, I'm giving you this initial sequence here. Will that be fine? Yes.
Yeah.
Yeah. In this way, you will be able to understand the topic in a more clear manner. Okay. So shall we start? Yes.
Yeah. Sure.
Yeah. Okay. So the sequence is fine, Sana. Yes.
Yeah.
Yeah. Okay, so this sequence I have made specifically for your class, so that because you will be able to understand it in a very sequential manner. Okay, if I give you haphazardly, it'll be confused. So sequential manner I'm going. So let's start with carbohydrates. So if I say carbohydrates, what are carbohydrates, Sana? Can you tell me?
They are
Yes, carbohydrates
uh,
are
organic compounds,
compounds of, yes,
hydrogen,
hmm,
oxygen, and carbon.
carbon, hydrogen, and oxygen. Carbon, hydrogen, and oxygen occur in different proportions. Different proportions. Very important. Different proportions. Okay. Carbohydrates are also called as sugars. Okay. Or we call them as saccharon. Saccharon meaning what? Sugars.
Mhm.
Okay, saccharon meaning sugars. Carbohydrate will have aldehyde group, aldehyde group, and will have a ketone group. Okay, it will have an aldehyde group or a ketone group. This will be present in the carbohydrate. Okay, it will present in the carbohydrate. So let's see here. The aldehyde group will be the ROH or RHO. Okay, and then we have the ketone group, right?
Yeah.
Okay, we have the ketone group, ketone functional group. Let us see here, or I'll put it in this manner. This will be better to understand. Let's see here. Okay, we have an aldehyde group HCHO, and we have a ketone group. Okay, we'll have a this is formaldehyde, sorry, CHO group. Okay, and then we'll have a ketone group. Is this clear? Yes.
Yeah.
Yeah, so we'll put it in RCHO and RCOR. That will be a ketone group. Okay,
Mhm.
I've made formaldehyde here. Let me put this R down. Yes. So aldehyde group and ketone group will be present. Carbohydrates are the most important substrates. They're the most important substrate which can provide an immediate, which can provide an immediate source of energy. This can provide an immediate source of energy.
Mhm.
Immediate source of energy, ATP if it is required. So we break down glucose immediately to produce pyruvate. The pyruvate in turn in aerobic respiration will lead to the formation of ATP. Okay, so energy, by referring to energy, I mean here ATP, which is produced in the mitochondrion. Okay,
Yeah.
so in the presence of oxygen, glucose will be broken down to produce ATP, even in the absence of oxygen also. But the amount of amount of energy that is produced in the absence of oxygen, that is less. Energy produced in the presence of oxygen, that is more. These points are clear, Sana?
Yeah.
Yeah. Okay. So aldehyde group, ketone, they're the
Mhm.
most important substrate. Now, carbohydrates, according to the properties, according to the units, they are classified into three major groups. Okay, they are classified into three major groups. So these three major groups we have is called as monosaccharide. Then we have oligosaccharide. And then we have is polysaccharide, oligosaccharide, monosaccharide, and polysaccharide.
Yeah.
So the word saccharon, the word saccharon tells it's a sugar. Okay, everywhere sugar. See, everywhere it's a sugar. But here specific words are given: mono, oligo, and poly. Let's go to mono. Mono means these are made up of single units of carbohydrate. But which carbohydrate we'll see? Single units, only one unit. Okay. For example, glucose. Okay. For example, glucose. Glucose is the simplest sugar. Fructose. Fructose is also a simpler sugar. Simplest. Okay. Monosaccharides are called as simplest sugar. Now, suppose, let's see here. Okay, so glucose, for example, glucose, these are termed as simple sugar. These are termed as what? Simple sugar. Okay, monosaccharides, they cannot be further hydrolyzed. They cannot be further hydrolyzed. Hydrolyzed means they cannot be further broken down. They cannot be further broken down. Okay, they are so simple. They are the simplest unit. They cannot be further broken down. This is an important point. We have occurrence of glucose in various components. Okay, glucose, fructose, all are there in various components. Okay, so let us see here. We will find glucose. Glucose is a blood sugar. It is the sugar that is present in the blood. Excess of glucose is removed by excess of glucose is removed by the hormone. Can you tell me which hormone?
Um,
Yes.
I'm not sure.
Can you tell me which hormone? Yes. Excess of glucose in our body is removed by the hormone insulin.
Okay.
If the glucose is excess in blood, it is removed by the hormone insulin. Okay, it is removed by the hormone what? Insulin. Is this clear?
Yeah.
Okay. Next. So let's see here. Now, when I talk about glucose, when I talk about carbohydrate, okay, glucose. So let's see another example. Okay, glucose. Glucose is also called as grape sugar. It is the only sugar which is found in grapes. Grape is a fruit, so usually it should have fructose, but the grape has what? Glucose as its sugar. Okay, glucose as its sugar. Glucose. Glucose in the presence of oxygen will break down to give pyruvate or pyruvic acid in the presence of oxygen. That means in aerobic condition. Okay, pyruvate. This pyruvate in the presence of mitochondria will produce ATP.
Okay.
Okay, it will produce ATP. All right, keep this in mind. Okay, it'll produce ATP, mitochondria. Aerobic, we call this as aerobic respiration. Aerobic respiration, glucose. Okay, now next, see, after that we have the next glucose, that is called, okay, next monosaccharide, that is called as fructose. Fructose is a cane sugar. Okay, it's a cane sugar. That means it is found in sugar cane. Fructose is also, sorry. One second. Fructose is not a cane sugar, sorry. Fructose, one second, is a fruit sugar, and it is found in fruits. This is also the sweetest sugar, fructose. Next, we have is galactose. This is the milk sugar. It is present in milk. Galactose is also referred to as the brain sugar because during the early development of the brain in the embryo, in the fetus, in the young ones,
Mm-hmm.
the galactose is required for the development of the brain. So it is called as the brain sugar. Clear?
Yeah.
Next, after this, the disaccharides, sorry, after monosaccharides, we have oligosaccharides. Oligosaccharides contain 2 to 9 units of monosaccharide. 2 to 9 units of monosaccharides are joined. Okay, so this is a monosaccharide, this will be joined. Okay, this is a monosaccharide, this is joined to another. So 2 to 9 units of monosaccharides, when they are joined, okay, to form is what we call as oligosaccharides. Now, this is one carbohydrate, this is another carbohydrate. So, how both the carbohydrate will join? They'll form a bond. They'll form a bond here. There is always a bond that will keep the two units together intact. And this bond, which is present between two monosaccharide units, is called as glycosidic bond. Okay, it is called as glycosidic bond. So
Mm-hmm.
the question comes, where is glycosidic bond present? Answer will be glycosidic bonds are present where? In the carbohydrates between the two monosaccharide units. So Sana, can you tell me glycosidic bond is present or absent in monosaccharide?
Present.
In monosaccharide,
Present.
glycosidic bond in monosaccharide,
Oh, absent. Absent.
absent, because monosaccharide is only a single unit of carbohydrate, so they don't need any bonds. Okay, oligosaccharide. Oligosaccharide can be dry, tri, tetra, many. So one of the most common oligosaccharides that we are going to study are the disaccharides. Disaccharide. That means two units of monosaccharides join to form a single unit of disaccharide. Okay, single unit of disaccharide. So let's see this. Okay, so disaccharides we have, let's see, the first one. We will have lactose. Lactose is present in milk, and the enzyme lactase will help to digest the lactose. There are people who are not able to digest the lactose. There are people who are not able to digest the lactose because they lack the enzyme lactase. Those
Mm-hmm.
people are said to be suffering from,
Lactose intolerance.
very good, yes, lactose intolerance. Okay, so lactose will be broken down in the presence of the enzyme lactase to form glucose plus galactose. Okay, glucose plus galactose. All right, next is sucrose. Sucrose is called as the cane sugar, and we extract sucrose from sugar cane. Sucrose in the presence of sucrase enzyme, or we also call as invertase enzyme, converts into glucose plus fructose. Okay, cane sugar. We get it from where? Sugar cane.
Mm-hmm.
Is this clear?
Yeah.
Yes. We have maltose in the presence of the enzyme maltase will be broken down to two units of glucose only, glucose plus glucose. Okay, glucose plus glucose. All right, these points are clear, Sana?
Yeah.
Yeah. Okay. All right. So okay, so we will have many type of disaccharides, and we are going to see on the disaccharides, we are going to see some specialized monosaccharides, all this. So before that, let's go and have a look at the polysaccharides. The polysaccharides are the larger carbohydrates. Okay, poly means large. Saccharide means saccharon. Large sugar unit is called as polysaccharide. Clear? Yes?
Yeah.
Okay. They are polymers. Polymers of sugar. They are what? Polymers of sugar. This point is clear?
Yeah.
Polysaccharides can be homopolysaccharides, which is made up of similar units, or it can be heteropolysaccharide, made up of different units. To understand polysaccharide, let us see the different types of polysaccharide. I'll bring this down here. So Let us see now the polysaccharide. Polysaccharide, as I told you, these are polymers of carbohydrate. Polysaccharides, okay, polysaccharides, many units. These are larger compounds, larger compounds. Okay, polysaccharide may be made by the condensation of many monosaccharide units joined together. Okay, more than 9 monosaccharide units joined together. They form what? Polysaccharide. Polysaccharide occur in various forms, and they're also one of the most important. Okay, so let us start with one by one polysaccharide. Okay, so uh, can you name one polysaccharide, Sana? And I will start with that.
Cellulose.
Yes, cellulose. Cellulose. Cellulose is a structural polysaccharide. Structural polysaccharide. Cellulose are found in the cell wall of plants. Okay, they're found in the cell wall of plants. Okay, they form the structure of the cell. That's why they are called as cellulose. Cellulose are made up of repeating repeating units of glucose. Okay, cellulose are made up of repeating unit of glucose. All right,
Yeah.
cellulose, they are linear, not branch. They are linear chain polymers. Polymers, single chain polymers. Okay, cellulose provide the fibers in the plant cells. Okay, fibers in the plant cell. Is this point clear?
Yeah.
Any doubt here?
No.
Yes, sure.
Yeah.
Yeah, okay. Next, when we talk about cellulose, when we talk about polysaccharide, we have the starch. What is starch? Can you tell me?
Um,
Where
it's
is starch found?
in plants.
Yes, in plants. Yes.
Yeah.
Okay. What is the difference? Okay, in plants, how are they? Can you name one structure in the plant where the starch is stored?
Um, it's
Yes.
stored in
Can you tell me?
chloroplasts.
Yes. Chloroplast. Yes. Starch are the storage polysaccharide. Storage polysaccharide in plants. Specify this. Okay, they are the storage polysaccharides in plants. Whatever carbohydrates are present in the plants that are converted into starch for the storage purpose, because plants can efficiently store starch for further use. Okay, storage. So starch can be of amylose, amylose and amylopectin. Starch can either be amylose or amylopectin. Now the difference is the amylose. is amylose is, okay, is a linear chain. Amylopectin, okay, amylopectin, it is what? It is branched, okay, it is branched, okay, clear?
Yeah.
Yeah, okay. Sana, you tell me, have you read about the bonds like alpha 1 to 4, beta 1 to 4?
Yeah.
Yes, okay. So today we are going to do the theory. So first you study the theory, then in the upcoming class we are going to do all the structures and the bonds of carbohydrates. Clear?
Yeah.
Okay. We have peptide bond, we have glycosidic bonds as well. Starch, these are the storage polysaccharides. In where in plants it can be either amylose linear chain or amylopectin. Okay,
Yeah.
starch. Then let's see here, cellulose you said structural polysaccharide, starch with storage polysaccharide. We have glycogen. Glycogen is a storage. Glycogen is a storage poly saccharide. It is a storage polysaccharide, but where? In the animals. In the animals. Glycogen are made up of linear, linear chain of glucose, linear chain of glucose. Okay, glycogen is the storage polysaccharide in animals. So glycogen is also sometimes called as the animal starch. Animal starch, because starch is the storage polysaccharide in plants, glycogen is the storage polysaccharide in the animals. Okay,
Yeah.
glycogen. Sana, can you tell me, in our body where is glycogen stored?
Um,
Two places.
is it stored in the liver?
Yes, in the
Liver?
liver. Yes, liver, and in the muscles. Okay, liver and in the muscles. So whenever in our body there is glucose, and our body, what happened, insulin will take that and convert it into glycogen and store this glycogen in the liver and store this glycogen in the muscle. Now, okay, so in the liver, glucose is converted to glycogen and it is stored. Now, sometimes in our body, the glucose level may fall down drastically. Then at that time, what we need to do? We need to search for sweet food, or there is a mechanism in our body. Now, whenever what happens in our body, the glucose level falls down. We need to maintain glucose level. Very important. Okay, very 120 to 140 mg per dilution of glucose should be there in order to maintain the blood pressure and all. So suppose, for example, in our body, the level of glucose is less. So in the liver, the glycogen, okay, glycogen is converted into what? Glucose. Okay, glycogen will be converted into glucose and can be used. Again, excess glucose is converted into glycogen and stored. This cyclic formation of glucose and all, this is called as Corey's cycle. Okay, what it is called? It is called as Corey's cycle. That means the glycogen or the starch or a glycogen that is stored in the liver, that is, okay, we can retrace it, we can reuse it, but the muscle glycogen is never retrieved. Muscle glycogen. is never retrieved. Clear?
Yeah.
Okay, muscles will use up all the glycogen when we do exercise. When we do exercise, now suppose for example, I'm continuously writing, sometimes my hands will pain. That is because my muscles have totally used up the glycogen. These points are clear, Sana. I'm doing only the theory now, so you
Yeah.
concentrate only on the theory. In the upcoming class we're going to do the structures.
Okay.
Yes. Have
Yeah.
you understood? Any doubt here?
No.
Okay. So glycogen. Glycogen is a storage polysaccharide. Starch is a storage polysaccharide. We'll come to another one which is called as inulin. Inulin is also a storage polysaccharide. Okay. Now, where not insulin, inulin, where is this found? These are found in the roots of dahlia plants. Okay, in the roots of dahlia plants. Inulin. Okay, inulin is very mild in taste, like sweetness, very mild in sweetness. All right, and it is, readily excreted by glomerulus. So, for example, suppose for example, if we go to the doctor, urologist, he will give a glomerular test. He will see how much inulin is there in our body. Okay, if it is there, how much inulin is there in the body? If more inulin, that means the kidney is malfunctioning, because normally glomerulus or kidney should eliminate the entire inulin, but here in the blood we have a huge amount of inulin. That means the glomerulus or the kidney is malfunctioning. This is a test conducted, okay, by the doctors in urology. Okay. Okay, roots of dahlia plants, inulin. Okay, it's a storage polysaccharide, roots of dahlia plant. Is these point clear? Yes.
Yeah.
Chitin. Chitin, just like starch is a structural polysaccharide, chitin is also a structural polysaccharide. Okay, structural polysaccharide. The chitin, this is found as an exoskeleton, where exoskeleton on the body of arthropods. That means we are talking about insects. Is this clear?
Yeah.
Okay. All right. Exoskeleton on the body of what? Arthropods. Okay. Now, chitin. Okay. Structural polysaccharide. Chitin. Chitin is a homopolysaccharide. Homopolysaccharide. It is made up of similar units. Okay, one more thing. Inulin is made up of glucose, many units of glucose. Chitin is a homopolysaccharide. It is made up of the similar unit. And what is that homopolysaccharide? It is made up of N-acetyl glucose amine. N-acetyl glucose amine, otherwise called as NAM. This is also found in the cell wall of bacteria. Okay, cell wall of bacteria. So chitin consists of N-acetyl glucosamine. So what? There are repeated, there are repeated units of N-acetyl glucosamine. Sorry. Uh, One second. Where it went? NAG, N-acetyl glucosamine. Repeated units of N-acetyl. So one, like one N-acetyl glucosamine, another bond. One N-acetyl, again another bond. Like this, many N-acetyl glucosamine will join to form a single chitin. And what is N-acetyl glucosamine? It is a modified glucose. This is clear?
Yeah.
Okay, one more thing on glycogen. Glycogen is highly branched. Okay, it is highly branched. So, Sana, till here, is there any kind of confusion? Yes?
No.
No? Sure?
Yeah.
Okay. So, these are the different form of polysaccharides. Clear? Yes?
Yeah.
Okay, different form of polysaccharides. All right, we have chitin, we have cellulose, we have glycogen, many different type of polysaccharides. Okay, so we have studied about disaccharide, the polysaccharide. Okay, then now, very important. Suppose, for example, let us see here. Okay, let us see here. Take for example, let me give example from here only rather. Cellulose. Herbivores or animals, they have the cellulose digesting enzyme. What is it called as? It is called as cellulase. It is called as what? It is called as cellulase enzyme. And these animals are able to digest the cellulose. For example, in the cow and all, okay, ruminant animal, they digest the cellulose in the presence of methanogens, a type of bacteria, and we use the cow dung to produce biogas plant. So animals, herbivores, depending on the plants, they have cellulase enzyme to digest. But we humans, we lack cellulase enzyme, so we cannot digest plant products. Okay, the main fibers cannot be digested. Clear? Yes.
Yeah.
Any doubt? Sana, have a look at the whiteboard. See whatever I have taught. Have you got any doubt? Clarify your doubt, please.
No, I don't think so.
Yes, all okay. Why don't you do one thing? Why don't you just, see your book? You have your book in front of you?
Yeah.
Yes. Why don't you just see the book on the carbohydrates, on polysaccharides, and monodisaccharides? And then if you have any doubt, or else we can proceed.
Okay.
Yes. Okay. Okay. Any doubt?
No.
No? Clear? Can I proceed then?
Yeah.
Okay. So we have done enough of carbohydrates. Let us do the next most important component of our body. And at last, we are going to do the lipids. Now, we are going to do is the proteins. Okay, we are going to do is protein. Now, Sana, can you tell me what is the importance of proteins? What is the
Um,
importance of proteins? Yes.
Proteins.
Yes, yes, tell me.
Growth.
Proteins are what? How can you possibly define a protein?
Um,
Yes,
they're.
how can you possibly define a protein?
I know they're important for growth or like
Sorry.
for repairing cells.
Yes, cell repairing. Yes.
Um, I know they're made out of amino
And
acids.
isn't proteins play important role in our body?
Yeah,
Yes. Hemoglobin. What is the function of hemoglobin?
to
Yes. What is the function of hemoglobin?
it's
To transport gases, right?
yeah.
Okay. In our body, metabolism occurs. Various enzymes that occur in metabolism are proteins.
Mhm.
Okay. So proteins play a very important role. So let's see. First of all, okay, what is protein made up of? The backbone of protein is none other than the amino acids. Amino acids are the backbone of protein. If you break down protein, you will get amino acids. Clear?
Yeah.
Okay. Next. Next, let us see. Okay. So amino acids, we call them as what? Building blocks of protein. The amino acids are joined together by a bond called as the peptide bond. Peptide bond. The amino acids, we have 20 different amino acids. Out of that, we have some of them, approximately 10. We call them as essential amino acid, and the remaining is non-essential amino acids. Non-essential amino acids. Essential amino acids are those amino acids which our body cannot synthesize, so we need to get them from the food that we eat. Okay, essential amino acids. Our body cannot make them, cannot synthesize them. So get it from outside, get it from the food that we eat. Essential. Non essential amino acids. Just because the name is non essential does not mean it's not important. It's important, but non-essential amino acids we can synthesize in the body. We do not need their supplement from the food, because our body has the ability to synthesize them. Point is clear, Sana? Yes.
Yeah.
Okay. All right, next, let's see. Okay, now amino acids. Okay, so let's see here. Amino acid have a central carbon atom. To this carbon atom, various groups are attached. We have the NH2 or the amino group. We have the COOH or the carboxylic group. We have the hydrogen, and then we have the R. The R stands for variable group. Many group, alcohol, hydroxyl, carboxylic acid, many things can come and attached. And whichever group comes and attaches to the R, it will decide the property of the amino acid. Is this point clear?
Yeah.
Yes, is this point clear? Okay, it
Yeah.
will decide the property of the amino acid. Now, to this carbon atom, to this carbon atom, all the groups are attached. So this carbon atom is called as the alpha carbon atom. What carbon atom? Alpha. So
Mhm.
at one end, we have an amino group, and on the other end, we have the carboxylic carboxylic group. Okay, amino group on one hand and carboxylic group on the other hand. So the group that so the pole where amino group is present, it's called as the N terminal. Okay, it is called as N terminal. C group is called as C terminal. N terminal and C terminal. All right. So this is what the structure of the amino acid is. Okay. When the amino acid, when the amino acid will ionize, when the amino acid ionize, we have an amino ion here and a carboxylic ion here. Okay, amino ion and a carboxylic ion. All right. So both positive, we will have the positive ion at this place, and we have the negative ion here. So amino acid or amino acid ion is that particular group which will contain, okay, the ion which will contain positive ion also and negative ion also. Okay,
Yeah.
positive ion and negative ion. Is this point clear?
Yeah.
Yes.
Yeah.
Okay. So yeah, Sana, can you tell me mm, positive ion, negative ion? Okay. So you tell me here at a particular time, will amino acid have both the ions or only one ion?
It'll have both of the ions.
Yes, they'll have what? Both the ions, right?
Yeah.
Yes. Okay, so this is the property, this is the ion weight of amino acid, which is called as Zwitter ion. Okay, Zwitter ion. Presence of both positive ion and negative ion in a molecule is called as Zwitter ion. And the Zwitter ion can be formed only in a particular pH. At a particular pH, what happens? The amino acids dissociate to form the ions, and it consists of at the same time positive and the negative ion. So that pH is called as the isoelectric pH. Okay, isoelectric pH. These points are clear, Sana? Yes?
Yeah.
Okay. Now, an amino acids. Let's see here. Um, as I said, we are going to go into the structure lately. Let me see here. Okay, we'll come to this protein later on. Okay. Now, let's see here. Okay, now see here. Uh, Suppose, for example, okay, and this ability of the amino acid to survive, this ability of the amino acid to survive, having both positive and negative ion, is called as amphoteric property of amino acids. Amino acids having the ability to survive both as positive and negative ion, M4. amphoteric property of amino acids. Okay, amphoteric property of amino acids. This point is clear, yes?
Yeah.
Okay. Now, how do amino acids form proteins? So when a protein is formed, many amino acids will join together, forming a bond called as the peptide bond. And that peptide bond, okay, that peptide bond will form the protein structure. Okay, form the protein structure. Now, the question is, we'll see here itself. Okay, suppose, for example, this is the structure of amino acid that I showed you. Now, for example, there is another amino acid coming to join this to form a protein. Now, the question is where the upcoming amino acid will join. So now you see here, okay, when a second amino acid comes, there is always a bond will form between the carboxylic group of the first amino acid and the amino group of the second amino acid.
Hmm.
Okay, they will join to form a bond which is called as a peptide bond. Peptide bond. Clear on this?
Yeah.
Yes.
Yeah.
Yes, okay. So many amino acids can join together to form a huge polypeptide chain. Polypeptide chain. And the polypeptide chain is none other than our protein. Okay, it's none other than our protein. Clear?
Yeah.
Okay. Anything till now you have not understood, Sana?
No.
No, all clear?
Yeah.
All right, let's see this. Okay. Now, now we know that, okay, Sana, can you tell me when are protein formed in our body?
Um,
When is protein formed in our body?
when
Yes. Think, think,
um,
think. When is protein formed in our body? What is that process called as?
oh, condensation reaction.
Condensation reaction? Not, it's a process, but won't be the appropriate answer.
Oh,
It'll take place there, but not an appropriate answer. Yes.
protein synthesis.
Yes.
Okay.
Okay. Whenever there's protein synthesis occurring, okay, a protein will be formed in our body. Okay, protein will be formed in our body. Now, you know mRNA, right?
Yeah.
Okay. So, where does protein synthesis take place?
Um,
Where does protein synthesis take place?
in cells.
Yes. Recall, yes.
Um,
Where does protein synthesis take place in our cell? Yes.
ribosomes.
Yes.
Okay.
Ribosome. Forgot.
No, no, no. Yeah.
Ribosomes are called as protein factories. It is in these ribosomes that protein synthesis occur. Okay, protein synthesis will occur. Clear?
Yeah.
Okay, protein synthesis will occur. All right, so protein synthesis occurs Okay, when protein synthesis occur, what happens? The amino acid will join to form a huge polypeptide chain. Okay, huge polypeptide chain. This polypeptide chain, first when it is formed in the ribosome, it will go to the endoplasmic reticulum, then to Golgi apparatus. Finally, it is released into the body. Clear?
Yeah.
Okay. Now, there are 20 amino acids. And these 20 amino acids, they combine in different manner to form this protein chain or polypeptide chain. Okay, so let's see here a few important things. Okay, so ribosomes are called as protein factories. It is in the ribosome that protein synthesis will occur. Okay, protein synthesis will occur. So now you see here, let's see this. Okay, all right. So once the protein is formed, a process called as protein folding is very important. Very important. Once the protein is formed, according to our requirement, according to the genes that is present, the protein folding takes place. Misfolding of protein, if the proteins are not properly folded, they lead to mutation, and mutation may be cancerous also.
Mm-hmm.
So appropriate protein folding is very much important. Okay, appropriate protein folding is very much important. So now let's see here. Okay, so based on that, we have different types of protein. We have linear protein. Okay, we will have linear protein. We have collagen helix, beta pleated chain. All right, we have the 3D image of a protein that we get in the enzymes. Okay, all these are nothing but how the proteins are being folded in our body. Clear? Yes.
Yeah.
All right. Now, one more thing. Okay. All right. So, when we eat protein in our body, okay. Now, see, when we eat carbohydrate, when we eat carbohydrate, we get what? Energy. Carbohydrate, as I mentioned, is an immediate source of energy. Okay, carbohydrate is an immediate source of energy. All right, then next, suppose for example fats. Okay, fat, if you had a diet which is rich in fat, so you may not feel hungry for a long period of time, because fats are also energy sources.
Mm-hmm.
Okay, fats are also energy sources. So now let us come to the protein. Okay, let us come to the protein now. Now protein are building blocks. One second. Amino acid. They are building blocks of protein. Okay, amino acids are the building blocks of protein. It is by joining of the amino acid with the help of peptide bond that protein or the polypeptide chain is formed. Protein is also called as what? Polypeptide chain. Now the protein will be folded in different manner. We have the primary structure of primary structure of protein, which will be linear chain. Primary structure is a linear chain of protein. We then have we will have the secondary structure. We will then have the secondary structure of protein. The secondary structure of protein can be, okay, it can be the helically coiled protein, like this right-handed coil protein. Okay, this is called as the alpha helix, and this type of helix is found in the hair, protein hair, that is the keratin. Okay, keratin, we have like this plate like structures. Okay, they form like this plate like structures. So do you know, Sana, what is it called as?
Mm, not sure.
No, it's called as beta. One second. It is called as beta pleated chain, and this is found is fibroin. Fibroin is the protein of silk. We wear no silk cloths.
Yeah.
Yeah, okay, so protein of silk. Okay, then another one will be the collagen helix, three helix. This is called as collagen helix. This is found in the collagen fibers, which is present in our body. It helps to form the new layers of skin, the fibers, collagen fibers. We have the tertiary structure of protein. The tertiary structure of protein is the only structure that will give a 3D image. That means it is the enzymes. It is the enzymes. The protein are being folded continuously, folded and refolded. They'll form a space like this. Okay, they'll form a space. This is called as the active site of the enzyme, where any other substrate can come and bind. Tertiary structure of enzyme is the only structure that will give a 3D structure.
Mm-hmm.
Okay, tertiary structure. Then we have the quaternary structure. These structure are found in the complex protein, such as hemoglobin. This point is clear?
Yeah.
Yes, any doubt?
No.
Okay, all right, understood, no?
Yeah.
Yes, these are the different structures of proteins. Okay, protein structure, very important. We have the primary, we have the secondary, tertiary, and quaternary. All right, and based on this only the proteins are folded. Now suppose, for example, when we digest protein, what we get is, okay, we will get amino acids. So when in our body there is excess amino acids, suppose for example excess amino acids, okay, excess amino acids are produced. Now we cannot keep this excess amino acid. It is not possible to keep this excess amino acid. We can't keep it. Okay, so excess amino acid, instead of being kept in the body, if they are kept, they will affect the biochemical reactions. So instead of keeping them in the body, we have to eliminate them. Okay, we have to eliminate them, remove them from our body. Okay, remove them from our body. So amino acids will undergo deamination. Deamination.
Yeah.
It's properly undergo deamination. Deamination. Clear?
Mm-hmm. Yeah.
Okay. After deamination, these are then removed. These are then, one second, after deamination, it is converted into, yes, can you tell me it is converted into
Uh,
ammonia, right?
okay. Yeah.
This ammonia will be converted into urea, and this happens in the liver cycle called as Ornithine cycle, which is occurring in the liver. Urea, urea is then eliminated in the urine. Clear?
Yeah.
Yes. Do you know the Ornithine cycle, Sana? Have you done it?
No, I don't
No,
think
you
so.
know any, just a little bit about Ornithine cycle? Yes.
Not really.
Okay. So see, you don't have to go much into depth. You just need to know that in Ornithine cycle, we have the ammonia. This ammonia will be converted into urea. Why? Because ammonia is highly toxic. It is highly toxic. Point number one. Point number two, it cannot be, it cannot, cannot be easily removed. Okay. Why? Because it is toxic. We need to break it down into smaller non-toxic forms. Okay, smaller non-toxic forms, only then we can eliminate it. So that's why ammonia will be converted into urea, which is Ornithine cycle, which is also called as urea cycle. And where it occurs in the liver. Okay.
Mm-hmm.
Ornithine cycle or urea cycle is under the influence of many enzymes. It's a many step process. And in this process, the ammonia will be ultimately converted into the urea. After that, from the liver sinusoids, the urea is dropped into the bloodstream, which will go into the kidney for elimination. This point is clear?
Yeah.
Yes. Any doubt here?
No.
Okay. All right. Now, see here. Proteins. Proteins are, very important. Proteins are very important. They form a very important part of our body. All right. Now, see here. When I talk about the protein, okay, when I talk about the amino acid, when I say amino acid, what is amino acid? Okay.
Mm-hmm.
Now, whatever the amino acid that will be formed in our body, this information is stored in where? In the RNA. Okay, RNA gets this information from whom? DNA. And where this information is present in DNA? In the genes. Clear or not?
Yeah.
Yes. Whatever amino acid will be produced, how the amino acid will be produced in sequence, how it will be folded, everything is governed by this gene which is present in where? In the DNA. All information is stored in the DNA. Clear? Yes. Okay,
Yeah.
all information will be stored in the DNA. So for example, let's see here one, we are talking about genes here. Okay, we are talking about genes. So what are genes? I can say that genes are nothing but segment of DNA that stores genetic information. Okay, segment of DNA that stores what? Genetic information. So that means what genes will be, what amino acids will be produced is governed by whom? The genes. Clear?
Yeah.
Yes, many genes will be produced. Okay, many genes will code for many amino acids. Many amino acids will join to form the proteins. Okay, will join to form the proteins.
Mhm.
So we have hemoglobin, which consists of alpha and two alpha and two beta chains. All right, these are polypeptide chains, two alpha, two beta polypeptide chains. All right, hemoglobin. Like this, we may have many innumerable, for example, proteins. So let us start with hemoglobin. HB is hemoglobin. It is the respiratory pigment in the body. It helps, okay, respiratory pigment in the body. All right, it will help in the in the transport of. Can you tell me? It will help in the transport of.
Um, the transport in
Yes, transport of gases. That is O2 and CO2. Clear?
yeah.
Next protein that we have, this is called as collagen protein. Collagen protein. These are found in the skin. It is found in the tendons. Okay, it's found in the tendons. Collagen is found in the walls of blood vessels. Okay, all right. So integrin, it's a membrane protein. It
Mhm.
is a membrane. It's a membrane protein. Okay, membrane protein. And these will help to communicate between the cell, between inside and outside the cell. They will help to, they will help to communicate. They will help to communicate between outside and inside the cell. Okay, outside and inside the cell. Lysozyme, lysozyme, which is given here. Lysozyme. This is a type of enzyme, a type of protein that will be present in the nasal mucus. It is nasal mucus. It will be present in the tears. Okay, it is present in where? It is present in the tears. It's present in the tears. Also, can you tell me where it is present?
Um,
Yes, also can you tell me where it is present?
I'm not sure where else.
Yes, it is present in the saliva.
Oh.
All right, it is anti lysosome is what it is. It is actually antibacterial.
Mhm.
Lysosome is antibacterial. Keep this in mind. It is antibacterial. Okay, that means what do you understand by antibacterial?
Um, no bacteria?
Yes, what is anti? What is anti?
Like against
Yes, against. And bacterial,
bacteria?
against bacteria. That means it is in the mouth. It will help to kill the harmful microorganism that enters our mouth, okay, that enters our body through the mouth. Is this clear?
Yeah.
Yes? Okay. All right. Antibacterial. There are various proteins. We have, okay, we have the plasma protein, albumin. Okay, albumin, different type of proteins will be there, different, so many different type of proteins. Now, proteins, very important, we have to understand that proteins at very, at high temperature, protein will undergo denaturation. What do you understand by the word denaturation?
Um,
What
it's
do you
like
understand by the word denaturation?
you alter the qualities, right?
Alter the qualities, okay. All right. What else?
Um,
Okay, I'll write here denaturation.
mhm.
Alter qualities. But first point, I'll not write alter qualities. Alter qualities. Yes. What does denaturation means?
Maybe like breakdown.
Yes, yes. That means when the temperature is high, what happens? The proteins will be broken down.
Mhm.
The peptide peptide bonds, they are broken down. Okay, peptide bonds are what? Broken down. Okay, peptide bonds will be broken down. So that is called as denaturation of proteins. I'll just give you one example here. Enzymes. We are going to study enzymes later on. Enzymes are also protein. At very high temperature, when enzymes undergo denaturation, that means the enzymes get what? Destroyed. Clear?
Yeah.
Yes? Okay. All right. So denaturation of enzymes. Okay. So now since we have done so much of protein, Sana, can you just recall what are the different functions of protein?
Oh, to
Yes,
help
different functions of protein.
the
Yes.
growth,
Sorry.
growth,
And
transportation, or
okay,
storage.
transportation. All right, growth and development, transportation of gases. Okay. All right. Then
Energy.
what about muscle contraction?
Yeah.
Muscle contraction. Our muscles, okay, our muscles have protein which is called as myosin and actin. Myosin and what? Actin.
Hmm.
Okay, these are contractile proteins. These will undergo contraction, okay, so that muscles can function. So muscle contraction, these are proteins. All right, so these are also muscle contraction. Then some of the proteins are hormones. They control various functions of our body, various biochemical reactions of our body. Along with our DNA, we always have a protein called as histone protein.
Hmm.
Always. Without the histone protein, there's no DNA. So histone protein helps in DNA packaging. Okay, DNA packaging,
Yeah.
histone proteins. All right. Then membrane transport. Okay, transport of components in the cell membrane we have studied, right? Intermembrane space. Sorry, we have studied the transmembrane proteins. Yes.
Yeah.
Yeah. So membrane transport. So when you see this note, you'll remember. I'll write here. comes membrane protein. This point is clear?
Yeah.
Transmembrane protein. Okay. All right. So these are some of the functions. Immunity, providing immunity. All right. So many are there. Important functions I've given you. Okay. Providing immunity. All right. Defense mechanism of our body. Packaging of DNA. All right. Various receptors on the surface of the DNA. Various hormones. All right. All of them. All of these are some of the important functions of the proteins. Clear?
Mm-hmm. Yeah.
Yes, okay. So, Sana, can you tell me what is the bond which is formed between the amino acids to form a polypeptide chain called as?
Um, it's the
Yes,
peptide bond,
sorry?
peptide bond or covalent.
Yes,
Okay.
peptide bond. Name the bond which is formed between the glucose molecules.
Oh, it's hydrogen bonds.
No, no, no. Hydrogen bond is water.
Um,
Glycosidic bond.
oh, okay, yeah.
Okay, glycosidic bond.
Mm-hmm.
All right. Okay. Now, sucrose is a monosaccharide or a disaccharide?
Sucrose is a
Monosaccharide or disaccharide? It's a disaccharide.
saccharide.
Yes. Okay.
Okay.
All right. So, Sana will end the session here. Okay.
Okay.
All right. So, have you understood? Yes.
Yeah.
Yeah. So, in the upcoming session in the next week, we are going to do more on the structures. Clear?
Yeah.
Yeah. Okay. All right. Then bye. Take care.
Well, thank you. Bye.
Bye.